Okay, so hello, my fellow Everthinkers. In this podcast episode, we were joined by Erik Frijlink, and he has been the head of pharmaceutical technology and biopharmacy at our wonderful university for the last 25 years, over 25 years. Along with that, he was also the head of pharmaceutical development at Solvay Pharmaceuticals, and so we walked around plenty of different landscapes, mostly academia, but we also took very many detours to the wild and wonderful world of the industry and how these two worlds differ, what we can learn as students from all the ideas that he shared from us. And it's been a wonderful experience.
I'm already very enthusiastic about this episode. Erik Frijlink is one of the very, very unique professors at our university. He has the unique combination between entrepreneurship, industry, and academia. And I'm very interested in how he ended up in academia, because he has a very, very unorthodox route towards to come to academia.
True.
You'll hear more about that, I guess, in the episode.
Exactly. Unorthodox for some, but for some I feel like even quite a, I guess, relatable one. But of course, we don't really have that much experience yet. So for us as aspiring academics, perhaps, it is a bit different, of course.
Of course, but it's certainly eye-opening and inspiring.
And his view on novel pharmaceutical kind of developments, how they can be administered, our little future section at the end of the podcast where he spoke about hypothetical developments and how the most important thing is actually identifying the issue at heart and then moving forward to try to create a technology to actually address the issue in people from a general kind of societal point of view. It was really fascinating to listen to. So I hope you enjoy this episode and
A great episode. Just listen to it.
Thank you. Professor Frijlink, thank you so much for being a part of this show. We're honored to have you. We would just like to start the conversation with a question we asked everybody that has come to our show. In your own words, just tell us a little bit about yourself.
Yeah, well, first of all, it's a pleasure to be here. What can I tell about Erik Frijlink? Erik Frijlink is a 62-year-old professor in pharmaceutical technology and biopharmacy. I worked at this university for more than 25 years as a professor. Before that, I was head of the pharmaceutical development at a pharmaceutical company, at Solvay Pharmaceuticals, which was an international position. I had departments in the Netherlands, in Weesp, close to Amsterdam. I had departments in Hanover in Germany and in Atlanta in the United States. Before that, I worked for a very short period, a little bit longer than 1 year, as a pharmacist in a hospital. And before that, I obtained a PhD at the University of Groningen and studied pharmacy at the University of Groningen. So I'm a pharmacist who followed a scientific career, more or less, with some industry in between.
Yes, fantastic. That's kind of what has, I guess, fascinated us about you. And specifically, what inspired you to pursue the field of pharmacy at the beginning, perhaps?
Actually, I did intend to study medicine when I came from high school. However, at that time, there were not sufficient places for all students to be accommodated in the medicine institute, and there was a lottery, and I was not in. So I had to find an alternative. And my chemistry teacher at high school told me, "Oh, there's a great alternative for you. You're good in chemistry, so please, you should consider pharmacy." So that was a reason to do pharmacy. I had in my family some people who were working in pharmacies or in pharmaceutical industry companies. So I had some ideas on what pharmacy was, and I thought it's an interesting and challenging field.
And how did your time during pharmacy shape your interest in pharmaceutical technology?
That came especially in the period when I performed my what's now called master research, and during my PhD, I did a PhD in pharmaceutical technology and biopharmacy, and also during my master, which was in pharmacokinetics, which is close to the pharmaceutical technology. If you combine pharmacokinetics and pharmaceutical technology, you get biopharmacy. So this is close to my heart. And I was intrigued by the challenge to improve the therapeutic effect of drugs through technology. So that was the reason for me to go into this field.
Very good. And were there other kind of pathways available to, I guess, pharmacy students, pharmacy graduates at the time? Or was this something, was research generally quite a rare occurrence for someone kind of finishing a pharmaceutical kind of degree or background?
It was certainly not rare. There were, let's say, at the time when I did my PhD, I think there was something like 50 PhD students in the pharmaceutical industry and in the pharmaceutical institute, sorry. And I think more than half, probably something like 70%, came from the study of pharmacy. So it was not rare. It was certainly not a majority. It was a minority who followed the scientific pathway, which it still is today. But in the end, we need pharmacists, and we need probably more pharmacists than scientists. So that was the reason that there is still a difference in the number of people that pursue a scientific career after studying pharmacy than people who become a general pharmacist, a community pharmacist, or a hospital pharmacist. There's a larger need.
Of course, yeah.
And did you have the vision from the start that this is the field that you want to specialize in, biopharmaceutical technology? This is the hot topic that you want to pursue for the rest of your life, or is this interest? Has this interest developed across time?
No, it really developed across time. When I studied pharmacy, I really found it difficult to make choices because I thought other subjects like the medicinal chemistry, like toxicology, analytics, they were also very interesting. As I said, I did my master's degree in pharmacokinetics. Pharmacodynamics and therapy are also interesting fields. So I was interested in many of these, and at a certain moment you have to make a choice, and that, of course, you make on, well, probably not always very rational grounds, but in the end I dropped into this field and I'm really enjoying it until today, because, well, it all has to do with making patients better, as I always call it. So the technology that we apply in pharmaceutical technology and biopharmacy is technology that has to do with the administration of active pharmaceutical compounds to patients. And if we can optimize the administration of the drugs, we can improve the therapeutic effect of a drug and reduce, for example, side effects, make therapy more convenient, safer. So altogether, it's really enjoyable to work in a field where you can have a real impact on the life of a patient. And that's actually what attracted me in this field, because you're actually connected to everything in pharmacy. I talk to the chemists who make the compounds that I have to formulate into a drug product. I talk to the people who are performing research and therapy to know something about the desired release profile of the people who do pharmacokinetics. I talk to people who are working in large-scale patient studies about the effect of a certain form of administration compared to another form of administration. So we're really like a spin in the web in technology in the entire field of pharmacy. In the end, you always have to administer the drug to the patient. That's the major purpose of what we are doing. So this is extremely challenging and shows the broadness of the field. It's really multidisciplinary. Of course, you're a specialist in your own field, but you also have to understand the other fields. If I don't understand anything about the background of asthma and COPD, developing inhalers becomes very difficult, and you may develop the completely wrong device. And that's something, and I cannot stress that enough, one of the nice things of pharmacy is that it's a scientific discipline which focuses itself on the application of the fundamental innovations from the sciences into pharmacotherapy, into making people better, into healing people. To understand the complexity of this, you need to understand how, on the one hand, the sciences bring their innovation forward, and you have to apply them in the real complex biological system the human being is. And that is really a science by itself, making this translation. So we do really multidisciplinary translational research, which in the end can have a huge societal impact.
Of course.
In addition to that, and that's extremely interesting. I think what we've seen, especially during the recent COVID pandemic, is that pharmacy, with its vaccines, had a huge impact on society. It was thanks to these vaccines that we were able to escape from the pandemic and get society started again.
Yeah, agreed.
Now I'm curious also because regarding this multidisciplinary approach, is it more flourishing within the academia than industry, or is the industry capacity for this multidisciplinary approach bigger? Because you also worked in industry right after after your PhD.
Yeah, no, I think they are the broadness of the discipline can be found both in industry as well as in the academia. In the end, the bottom line is very simple. Without approaching the challenges from multiple sides, you will never be able to develop a new drug, to add anything to pharmaceutical sciences, because it's only through the multidisciplinarity that you can understand the complexity of what you're doing and develop the appropriate products or therapies that you're working on. So in the end, the multidisciplinarity is everywhere in the field.
Yeah. And is academia kind of the perfect place, at least from your career, that you have kind of experienced this multidisciplinarity? Is it the best place to kind of see this collaboration in action, or was industry also a very good alternative where you had this element?
As I said, you see it in both. You learn different things in industry and in academia. In industry, you can see the entire picture because you have to really to go from start to finish, and you, as an industry, you're part of huge teams, people developing new drugs, and you really see everything. And it learns you how you can have a societal impact by bringing new drugs on the market and which actions you have to take for that and how complex that is. In the academia, you are sometimes a little bit more focused on your own field because, well, that's what you are appointed for, that's what you're working for. But on the other hand, as long as you don't have the other aspects in the back of your mind and take them into account, you cannot perform the appropriate research. You can probably come up with a solution, but then you end up with a solution looking for a problem, whereas the starting point should be your problem and finding an appropriate therapeutic solution for that. And so you need the multidisciplinarity also in university. I saw it more in the industry, since there also you have the business aspects and the approach of doctors and those kind of things. There's the economics in there. All the different steps that you have from basic pharmacology, chemistry, up to large-scale production and selling products. There are, of course, a couple of those things you don't do in academia. We don't produce drugs. We're not selling them. We sell the patents on our inventions. That's what we do. So that's a bit different.
Yeah. And so was this added aspect, were these extra kind of layers that are added to this multidisciplinary kind of act of actually bringing a drug onto the market, was that what initially inspired you to maybe move out of academia for a little bit when you became kind of the head of Solvay Pharmaceuticals?
I was not the head. I was only the head of the pharmaceutical development.
Of pharmaceutical development. Yes.
The development
so not
part.
head of the top,
no, I was not the director of the company.
Yes.
Don't worry. I thought, even when I finished my master's degree, or as it was called then, my doctoral, I already had the idea of going into industry, looking what's happening there, learning a lot. But then I was pretty young and I enjoyed research during my master, so I went into the PhD. And after the PhD, I thought, okay, it's good to see how industry is working. I liked working in industry a lot because of the additional things you could achieve there. The enormous collaboration that you had there, because in industry, in the end, you have to perform. You have to sell your drug. You have to bring your drug on the market. You have to sell it. So people are forced to collaborate, which is really different from university. University, you see more that investigators compete with each other. In industry, within your company, you are collaborating. Collaborating is probably the most important aspect of what you are doing. Otherwise, you cannot benefit from the advantages that the multidisciplinarity brings. So you learn a lot about how to collaborate, how you should do that, how you can work in multidisciplinary teams, how you use the power of a multidisciplinary team in bringing a drug to the market.
Your journey to professorship is quite unique. You went to the industry first, and then you came back to professorship, to
That's
academia.
a bit odd, yeah, sure.
Yeah, how did you manage to do that? Because people usually dedicate their whole career to PhDs and postdocs in order to obtain this position. How did you manage?
Yeah, well, I didn't manage it. I just dropped into it. Actually, after finishing my PhD, I went into industry. And I certainly had not the idea to become a professor after that. I had the idea of going into industry, have an industrial career. And actually, I was on my way with that. And then at a certain moment, they asked me here in Groningen whether I would like to become the successor of my previous PhD supervisor. He was going to retire, and the position was available. And they asked whether I had interest in that position. And so I thought, well, could be interesting too, because, well, there are differences between industry and academia. They both have advantages and disadvantages. In industry, there's always enough money. In academia, you always have to fight for your money, but you have a little bit more freedom. When in the industry, the board of the company tells you, well, we're going to stop this project. You have to stop, whether you like it or not. I had to stop projects that I really liked we were doing, but they were economically probably not feasible. Well, then you have to stop. So in academia, I could probably more or less do what I'd like to do. So at a certain moment, while working at Pharmaceutical, I was asked to become a professor here, and I decided to take that position.
But I could also imagine that maybe your work in industry has also contributed into some publications or some research lines that ended up being important in the academic world.
Yeah, sure. Many of the things that I learned in industry, I could apply and I could benefit from later in my academic career, especially the knowledge I obtained on how to develop drugs. I applied it when I was developing my inhalers or the new vaccine or oral products we worked on. So the research was more or less highly influenced by what I had seen in industry and my experiences there.
Mm-hmm. And did the research within industry work the same as academia? Did you also have publications and research papers that are published and have impact factors and stuff like that?
No, it's completely different. In industry, you are judged, you're evaluated on what you add to the development of a certain product when you're in R&D. So rather, you don't publish because there may be patent issues, etc., etc. So publication is not, as long as you're not in marketing, publication is not important in industry. So applications for projects, no, you don't do, because you work on the projects which the company works on. And sometimes when you're a project manager, you can, of course, propose new projects to the board. But there's no such thing as applying for a grant for a project in industry. That's not happening there. Once the board decides, okay, we're going to develop this drug, you're going to work on that drug. And you're evaluated on your performance in the development or the research on that specific drug. Did you bring that further? Were you able to make steps? Could you develop an adequate dosage form for it or a production process? Those are the kind of things that you're judged upon, and not on the academic things like publications or impact or lectures that you give. It's completely different.
Interesting. And so now that when you became a professor and you started your academic career, what grabbed your interest? What was the first thing you said, I wanted to investigate this?
Well, we had a couple of things were already going on in the group where I came. They were already performing some inhalation research. Anne de Boer was working on that. I was myself very much interested in the work with biopharmaceuticals, with the therapeutic proteins, vaccines, and more advanced therapeutic constructs, and how we could administer them, their stabilization, their behavior after release from dosage forms. So I was intrigued by that, and I was interested in targeting of oral products, so orally administered drugs, and how can we optimize the administration of those products. And well, there were of course a lot of other things I could have chosen to perform on, because pharmaceutical technology is quite a broad field. It goes from eye drops to suppositories to creams to tablets to inhalers to whatever you like. So it's quite a broad field, and of course you have to focus. I think it's very important to focus on the things which you find interesting, because that makes the work fun. And on the other hand, to focus on the things which are really challenging, which you consider a challenge, and where you have new ideas which can bring an innovation.
Exactly.
And speaking of, I guess, finding that focus, a large amount of your research has focused on inhaled dosage forms.
Yeah.
So what specifically in that field did you see could be improved, or did you have like immediately ideas while realizing, okay, at the university we are researching this in detail, what can I contribute? What was the kind of the first thought?
No, it was certainly not that from the start we had ideas how to improve. Of course, in collaborating with the people in my department. You never do things alone in pharmaceutical technology. So the department is much more important than I am. Please be aware of that. So when I say I had the idea, no, I never have an idea. Within the department, we have ideas together. We form our ideas together. So in discussions, you find challenges, you find ways to improve the pulmonary administration of drugs. And actually, the starting point in general will be, what are the challenges that are currently faced by the doctors and the patients in pulmonary drug administration? And then you can start to think about strategies and methods, technologies to solve those problems.
Okay.
And one of the things that we had identified was that there is a huge variation in the amount of drug that enters the lung upon inhalation, due to variations in the inhalation flow rate generated by patients. You see that there's more or less drug entering the lung and become therapeutically active. Well, that has a huge effect on the therapy, of course. So we started to develop inhalers in which the lung deposition, as we call it, the amount of drug that was deposited in the lung, remained more or less constant, irrespective of the inhalation flow rate, irrespective of how forceful a patient was inhaling the drug. Well, that was one of the problems that we identified. So it's always important, if you want to have an impact, to find the real life problems. Yeah. So we have to look into clinical practice. So that's what you have to do. It's often forgotten because people so much focus on their solution that they forget, hey, which problem are we solving? Yeah. So you should do it the other way. Start working on a problem and how to solve the problem.
So I'm thinking back to the point that you have mentioned about the inhalators, that the flow rate is independent or could be independent of the respiratory power of the patient.
No, the flow rate is not independent. The flow rate always is dependent on the energy generated by the patient, on the inspirational force that is generated by the patient, but the amount of drug that is deposited at a certain flow rate, that's what we made more or less independent from this flow rate. So there's always variation in flow rate. Patients have individual flow rate. If we look at us three here, we all, if I tell you, please give me a forceful inhalation, so a specific resistance, if I ask it you or me, there are different outcomes.
Yeah.
So the flow rate will vary. But thanks to the technology we developed, we were able to make the lung deposition constant, independent from flow rate. So that's the trick. Independent from how forceful you inhale, you always have the same amount of drug in your lung.
And I'm wondering, how do you do that? Do you, for example, do you have another energy output like a battery or something like that?
No, I can show it to you. We do that through a so-called cyclone. What we actually have is in our inhalers, this little piece. It's here. This thing.
Yes.
And what this is a so-called cyclone. And what's happening in here is this, that in here, agglomerated powders are brought if you inhale. So this is the entire inhaler. And if you inhale like this, if you inhale, then powder is sucked from a container here through this cyclone. And if you inhale more forcefully, then the effect of the cyclone becomes bigger with regard to the deagglomeration of the powder. So you get more fine particles. So then you would say, hey, wait a minute. When you talk about inhalation, the entire trick is how do I get sufficient fine particles in my aerosol? Because you bring particles in the air and that's what you inhale. And by inhaling fine particles, you bring drug into your lung. That's the entire trick. And fine particles can enter the lung quite well, whereas the particles that are too big will remain in your throat. However, if you inhale more forcefully, particles will always also be retained in your throat because the inertia of the particles is so high that they fly out of the band and stay in your throat. They don't reach your lung. So what you have to do now is that you find a system with which you generate more fine particles if you inhale more forcefully. And that's what the cyclone does. If you inhale more forcefully, you get more fine particles. Then you can lose more in the throat while still getting the same amount of drug in the lung. Because due to the higher speed, more particles fly out of the band. But since we started with much more fine particles, because of the cyclone, the amount of fine particles that entered the lung will always remain the same. So we all of a sudden had a system which gave us always the same lung deposition. And we have shown that in tests. Tests with the device were done, and we showed that we had something like a 30% variation in lung deposition. You say, oh, 30%. If we compare that to previous inhalation devices, they had 300%. So we were quite somewhat better.
So if I understand it correctly, so you have the cyclone, and the solution here is that if we have a stronger, if we have faster particles, then
if
we have
inhale more
loss.
forcefully.
Yeah, if we have faster particles, we have more loss, but then we try to start with an initial bigger amount.
Yeah, that's actually what you do. It's just compensation for the additional loss in the throat. So if you say, okay, it's the size range of 2 to 5 micrometer particles, which is extremely small. It is about 10 times smaller than baking sugar. So this is really small particles. So they tend to agglomerate. And what you do in the inhaler is to break up the agglomerates into nice 2 to 5 micrometer particles because they can enter the lung. Normally you have a certain fraction that turns into this size range. However, if we're now going to inhale more forcefully, we get more of those particles. Then there's of course the risk that they would also enter the lung. But they won't, because if you inhale more forcefully, your rate increases and you lose more in the back of your throat. So you compensate for the additional loss by generating more fine particles in the cyclone upon the more forceful inhalation.
Yeah, wow, very good.
And this sounds a bit counterintuitive, because you would say, okay, please generate always the same fraction of fine particles and you have always the same amount of drug in your lung. No, that's not the case, because if you inhale more forcefully, you lose more in the back of your throat. And we compensate for that loss.
But the cyclone seems like a very small structure. How do you manage to do that with this structure? What was the idea?
That was the design work by Anne de Boer. And it looks like Lego, but it was years of design work. And let's say finding the geometry of this part really was a hell of a job for him.
Wow.
Of course, you should balance the inhalation forces of the patients, the powder types that you have. We worked with so-called adhesive mixtures where you have large carriers and small micronized drug particles. How do they function? How do you break these agglomerates up? What kind of forces do you need for that? So that determines the diameter of the system. Then there is an aspect regarding the size of the inlet and especially the outlet. All those different aspects determine the performance of such a cyclone. So there was a lot of trial and error, calculation, mathematical approaches, modeling, which came up later. We did a lot of modeling, for example, on the third generation of inhalers that we developed, the Twinser, that has 2 cyclones in it, and we also did modeling work for that. So, of course, the research changed too in time. This was the first generation. Over there you see the second generation, the Genuair, the white one with orange. That's the second generation
It's the first
inhaler.
generation, or?
It looks exactly the same as the first one, but the inside is different. You cannot see that, but the inside is really different. And I cannot tell you what the difference is, because there are patents on that.
Oh, wow,
Yeah?
okay.
So the research changed, and then we come up with the disposable ones, which could work with much larger amounts of powder compared to these one. And there are some other examples of that too. Then first we started with 2 cyclones. Later we went back to disposable system with 1 cyclone. So we call that the Cyclops.
Cyclops. Well, okay.
Yeah, that's why.
Greek mythology.
Yeah. So this is the Twinser. The other one is called the Cyclops. And they all perform different, and they are more or less suitable for different micronized powders. And what probably also is important, people often think, oh, this is a disposable one, that's not good for the environment. But it's fully recyclable since it's polycarbonate. Yeah, so
it's good in some aspect also.
Yeah, if you just collect the used material, we can reuse them again and make new inhalers out of them. So it's pretty environmental friendly. We had the same environmental friendliness in this concept, because many inhalers, you just use them and throw them away afterwards. With our inhalers, that's not necessary, because there's a cartridge with powder, and you just replace the cartridge, and you still use the inhaler. So you can do a couple of cartridges, and then after 3 or 6, replace the inhaler. But it saves a lot of plastic, as you can imagine. Whereas with this one, we don't have plastic waste.
Yeah. These are all dry powder inhalers.
Yes.
And so could you maybe explain very quickly to the audience what are dry powder inhalers?
Well, dry powder inhalers are devices that are used to make people that need drugs in their lung for whatever reason. In most cases, it will be a pulmonary disease, asthma, COPD, or a pulmonary infection like tuberculosis. In that case, you have to inhale drugs. And there are different types of inhalers, and the dry powder inhaler is one of them. Dry powder inhaler is a system which allows us to generate an aerosol, a cloud of particles which we can inhale. And by inhaling this cloud, we get drug into the lung. And the dry powder inhaler is the device that can do it. In a powder deagglomeration principle, like I showed you here, the cyclone, we can generate such a cloud of fine particles upon inhalation. And the nice thing of a dry powder inhaler compared to many other devices, like for example the pressurized meter dose inhaler, is that it generates the cloud upon inhalation. So you don't need advanced or difficult hand-lung coordinations. You just generate the cloud while inhaling, because it's the energy from the inhalation that is used to generate the particle cloud.
So this is an optimization over the liquid inhalers, right?
Soft mist inhaler is the third generation.
It's third generation, was the 2000s, and this in the 1960s, if I remember correctly.
Yeah, well,
right?
the first ones were from the 70s, I think. But this is an optimization, and the last one was far in the 2000s. So we're optimizing, and we need all different types of inhalers, but some have advantages over the other ones. And so far, the dry powder inhaler is the one which provides us with the highest lung deposition. So for the majority of patients, the dry powder inhaler probably is the best way to inhale the drugs. But there are a few patients who cannot handle a dry powder inhaler, should be helped with a soft mist inhaler or with even a pressurized metered dose inhaler still. So there are different inhalation devices, and we need all of them. We even need the classical nebulizer, which is a device probably from the past century. But when optimized, it can still help some patients. For example, when there are no alternatives on the market for some drugs. Whenever we have a solution of a drug, we can nebulize it. Almost always, not always, but in the most cases we can. So when there's not a dry powder inhaler or pressurized metered dose inhaler, in those cases we could still use the classical nebulizer. For some drugs that's necessary.
So we left off about the topic of dry powder inhalers, and we know that one of the applications is antibiotics. What is really the contribution of dry powder inhalers to antibiotics?
The application of dry powder inhalers for the administration of antibiotics, of course, refers to their use upon pulmonary infections. Sometimes people have serious pulmonary infections due to a serious COPD, or, for example, an infection with tuberculosis. Multi-drug resistant tuberculosis is becoming a bigger and bigger problem, especially in the third world, related to the HIV pandemic over there. So what can we do if people have a pulmonary infection is to bring the drug immediately, the antibiotic, immediately to the site where the bacteria are. So with the pulmonary infection, inhalation really makes sense because it gives us much higher concentrations of drug at the site of action in the lung. So in this way, we can treat patients much and much better. And we can reach exposures which are sometimes over 100 times higher than after systemic administration by oral or by injection. So that's quite a difference, and that's extremely important since, for example, if we realize what's happening when a bacterium is resistant to drugs, or even with antibiotics, we have multi-drug resistant antibiotics, then that actually means that the bacterium, the microorganism, the mycobacterium is 10 times less sensitive to the drug. If a patient has an infection with a multi-drug resistant tuberculosis mycobacterium, in that case, we can inhale the drug and the concentrations are 100 times higher. Since the mycobacterium was 10 times less sensitive, we still have a factor 10 overshoot to kill the bacteria. We can rapidly, more or less, sterilize the airways, which prevents the transmission of the disease, since the disease is spread through coughing out little droplets containing the bacteria. If we now kill all the bacteria in the sputum in the upper airways, well, you can cough out any liquid you like, but there are no bacteria in it since they were killed by the antibiotics that you have inhaled. And you can only achieve that through inhalation, because if you take tablets or take injections, it takes days or even weeks before the airways are sterile, and people cannot transmit the disease anymore. So apart from treating the patient, which may be successful or not successful, at least we prevent transmission of the disease, the spreading of the disease into the community. And that is, in the case of, for example, multi-drug resistant tuberculosis, probably even more important than just treating the patient, which of course is also important. Understand me well, but this is more important.
Okay. And epidemiologically speaking, I assume this approach also, perhaps, I don't know if anyone has done a study, but to my understanding, it would perhaps help with antibiotic resistance as well if you are able to more effectively treat the lungs. Less basically antibiotics, I assume, are generally then consumed by the patient or for a shorter period of time. So
anyway, the faster you kill the bacteria, the lower the chance of the generation of resistance.
Of course.
So already from that basic principle, a rapid decrease in bacterial numbers is extremely important, and we can achieve that through inhalation in the pulmonary infections.
And has this idea been put in practice so far? Is there any clinical trials that we know of? Okay.
We have run clinical trials here in Groningen with a group of Onno Akkermans and Huip Kerstjens. They were involved in that. We have a huge tuberculosis clinic in Haren, in Beatrixoord, and we collaborate a lot with them. And we have a couple of first-in-man studies with new antibiotics and the Twincer or the Cyclops inhaler. So those are studies that we are performing, and we are developing new antibiotics for inhalation because we believe next to the normal tuberculosis therapy, the inhaled antibiotic can really add something in preventing the transmission and the spreading of the disease.
So are these inhalers going to replace the oral therapy completely?
No, it's not replacement.
Okay.
It's add-on therapy.
Okay.
So especially in the first period of the disease, immediately after diagnosis, we can rapidly decrease the bioburden in the sputum, in the upper airways, and that's really important. But after the equilibrium has established with the oral or the parenteral drugs, you don't need the inhalation any longer. But especially in the first period, this makes sense.
Interesting.
And sometimes probably you could, if you have highly resistant mycobacteria, you could continue to use the inhaled products. But that's the next step. And as I said, we're in the beginning of development. So there's still a lot of work to do. So we have quite a good job security, to be honest.
I guess. And talking about on-site action, I think also another very important application is the vaccination in the lungs.
Yeah, that's one of the subjects we have been working on a long time, vaccines. Not only inhalation, actually we started with stabilization of vaccines. I think, it's more than 20 years ago now that we started to stabilize vaccines. As you know, vaccines are complex biological molecules which are likely to degrade, and we were looking for ways to stabilize them. We worked with sugar glass technology, which means that we embed the vaccines in a amorphous sugar, solid material, and due to the interaction and with the sugar and the immobilization by the sugar, we can really stabilize the vaccines. For example, we showed that we could make influenza vaccines which were stable for 3 to 5 years at room temperature, whereas the same influenza vaccine in its liquid, as it is normally sold in the fridge, is only stable for 1 year. And we even have longer stability data.
So these are
nowadays.
dry vaccines?
These are dry vaccines. You just have to add water, dissolve them, and you can inject them again.
And since they are dry vaccines, we could also maybe use the dry powder inhaler.
Yeah, that was the logical next step then to make.
That's very interesting.
Because we had these dry powders, we thought, okay, the injection is nice, but there are also major disadvantages connected to injection because, you need medical trained personnel. In poorer countries, they use needles over and over, transmitting diseases like hepatitis B and those kind of things. So we thought, how can we find alternative routes of administration? Oral route could be possible, but certainly the inhalation route, in our opinion, was of interest. And the reason for that is that if you look at how we get some of those viral diseases, it is through inhalation of the virus. They're transmitted by the air, like the tuberculosis, which is a mycobacterium, but also diseases like influenza, respiratory syncytial virus, corona, of course we all know that. They're all spread because we inhale those particles. And actually the immune system in our lungs is completely tuned to treat those infections. So if you want to generate an immune response anywhere in our body, why should we do it by a subcutaneous or intramuscular injection? That doesn't make sense. There is not an immune system that is trained in my arm or in my leg. No, it's in my lungs. So bringing the vaccine into the lung to elicit an immune response, that really makes sense. Yeah. Because you may expect that you get a larger immune response there. And we have worked a lot with the influenza vaccine, and we could, for example, show that upon inhalation, we really got an increase in the immune response that was generated by the vaccine. Quite recently, a study was published with corona vaccine, came from China, where they also tested an inhaled corona vaccine, and they showed, actually, if you look very well at the data, an increase in the efficacy of the immune response. There was a larger immune response upon inhalation. So it makes sense to inhale a vaccine. And if you combine the advantages of making the vaccine dry, stable, and the increased immune response that is elicited after inhalation, of course, that's a highly interesting way of vaccinating people. Of course, this is not the case for all viruses. There are also viruses that are not transmitted through the air. For example, through mucus or through blood-blood contact, those kind of things. Well, with those diseases, probably the inhalation is not the appropriate way to administer the vaccine. Probably there you better do an injection still. But for those diseases which are airborne, as we call it, which are transmitted via the air, for those the inhalation may be a better way of administering vaccines than the injection. And of course it's much easier and cheaper. Now we stand in October in a row at our general practitioner because he has to give us the injections with the flu vaccine or I think now, and in the future also the corona vaccine. Then you can send the people even such a little inhaler they can inhale, and you're done. You're ready. You don't have to go to the doctor even anymore. Of course, you need to understand a little bit how you should inhale, but you can put that on a letter with some drawings with it. And in general, people may understand how they have to inhale. So then you can do a very fast vaccination campaign and a lot cheaper. Now we have to pay the doctors. You don't have to do that anymore then.
But out of curiosity, we talked earlier about the particle size and how the cyclone would have fine particles. But I can imagine that the vaccine, especially if we are using attenuated vaccines, the size would be bigger and maybe No,
it's not. It's not over 2 micrometer. No,
it's the same principle.
It's exactly the same principle. The vaccines that we use are much smaller than 2 micrometer. And 2 to 5 micrometer was the size of the particles that we have. So the solid particles that we use contain the solid particles that we use contain the vaccine in a carrier, for example, a sugar glass. And those particles have a size of 2 micrometer, but the particles of the vaccine, they're much smaller.
Okay. Yeah.
So that's not a problem.
Yeah.
If that would have been the case, we could have used the inhalation. If, let's say, the vaccine would have had a size of 20 or 50 micrometer, probably inhalation was not a suitable route, because then we couldn't get into the lung.
Yeah.
These particles were too big.
So these work perfectly well at this point.
This works really good. Yeah.
Then why don't we have these vaccines on the market? Why didn't we do it for corona?
Well, it's new.
Okay.
Although it's not new, because already by the end of the 60s and the beginning of the 70s, there were groups in the United States, Waldman and Haig, they already tested influenza vaccines. And that was highly interesting. They found really great effects. Again, they found that inhaling the influenza vaccine really protected the people better with less side effects than injection did. So it was interesting. But at that time, they did not have the appropriate technology for large-scale vaccination. There were only nebulizers or pressurized meter dose inhaler. Well, the pressurized meter dose inhaler, due to the propellant, is not suitable for a vaccine. The nebulizer is, but taking a dose with a nebulizer takes you probably something like 15 minutes. And then a nebulizer is extremely expensive piece of equipment. Well, that's not suitable for a large-scale mass vaccination campaign. Mm-hmm. That's not feasible. Yeah. If you have to take 15 minutes for every person that's going to be vaccinated, that won't work. Mm-hmm. So that was one problem. And on the other hand, they were working with the liquid, because for nebulizer you need the liquid, which was also not so interesting because it's not stable. Mm-hmm. So the technology that was necessary to make the inhaled vaccination work was not available. Nowadays, and that also takes time, because as I said, we're working on the stabilization of the vaccines now for something like 20 years. We're working on the inhalation probably something like 15 years nowadays. They also need time, but we now have the technology. If you take a disposable inhaler like the Cyclops or the Twinser, then you can administer the vaccine in 20 seconds. Oh. That's sufficient. And it's a single use like a syringe is. So it's more or less the same principle in that regard, but you have a more efficient vaccination, and you have a stable product. But those are all developments from the past 20 years, and not something that is already there for ages. And then, of course, meanwhile, the industry has focused on optimizing the injection route of administration. So especially for the already existing vaccines, they are not likely to change because they work.
Yeah.
Mm-hmm. And why should you change then?
Exactly.
So and I can understand that, although it's a bit disappointing. We think our inhalation is better. Yeah. But now with corona, probably there are new chances. If you look at the study from China, which was a somewhat larger study, you really see advantages of the inhaled vaccine over the injected vaccine. So that's a reason to continue working for new vaccines, and probably also for something that's going to be new in standardized or frequently repeated administrations like corona, to develop the dry inhaled vaccine and not any longer the injection.
Okay. So disrupting the current status quo is proving a little bit of a challenge.
That is really difficult. And I can understand that when you are a general practitioner and you have a patient and you're treating him years and years with the same drug, and he's doing well on that, there's no incentive to change.
Yeah. Yeah.
Even if you would know well, there are certain advantages, but your patient is happy, therapy works well, why would you change? I understand that GPs don't do that because then they're under risk of something going wrong and they have an angry patient in front of them. Well, that's what they don't like, as you understand. So you really have to focus on the new applications, the new vaccines here.
But isn't there also a financial incentive? I mean, the company that would take on this innovation would also have a more financial income because it's a new innovation with some
certainly. But that same company also has to make a big financial risk in developing the entire system. And it's expensive. Developing things with biopharmaceuticals, with vaccines, is really something that is expensive. And there's always the risk of failure. So you take a huge risk in starting to develop something. In the end, you can profit from it. But there are not that much companies that have a well-working product on the market to take this next step. Because there's always a risk related to that. I can give you one other example. At a certain moment, for a specific company, we developed an inhaled product of an existing drug. It was an antifungal drug. Well, there are fungal infections occurring in the lungs, so inhalation makes sense then. But then, at a certain moment, before we went into the clinical trials, the management of that company took the decision not to continue because they were afraid that new side effects may turn up just through the inhalation or through the fact that you did the study anyway, which would jeopardize their market, which was over a $1 billion market a year. So that's a risk, of course, for the company.
Yeah.
And they didn't want to run it. So initially they said, oh, it may be interesting. We can get some better therapy, more effective therapy, again, get additional years of patent protection. But on the other hand, the risk was that the current market would be destroyed if they would find in the new clinical study an unexpected side effect, which probably did not per se have to be related to the drug or maybe been caused by the inhalation or anything else. But still, it's a new side effect, and that jeopardizes the over $1 billion market. Well, that's something you don't want as a company.
Wow.
So
So it's a conservative industry in that aspect.
True. So like in life, everyone fears change to a certain degree.
Yeah.
Yeah, and the pharmaceutical industry definitely does at the same
They're
time.
really change avoiding.
Yeah.
But okay, it's understandable. And of course, when there's a tremendous advantage, they're going to change for sure. The mRNA vaccines probably 5 years ago were considered to be a strange niche for some nice researchers, but very little chance that they would ever become available or be used in large vaccination campaigns. Well, one pandemic later, we know better.
Yeah, exactly. So, well, hopefully we don't need pandemics to kind of push this technology forward.
No, certainly not.
But exactly.
It's a good example of how that changes the view, also within the companies.
Of course, yeah. Fingers crossed that, but such crazy events don't need to cause these risks, I guess.
Yeah, but maybe your career in industry and academia is a great example of how the duality of a scientist, entrepreneur could work out. And I'm wondering, maybe instead of these big industry, we could also have the environment of startups within academia. That also could maybe minimize the risk, right? Or not? Yeah,
well, actually, startups are used by the large pharmaceutical companies to minimize their risk in R&D. And what you see today is that real innovation comes from the startups, from the small and medium-sized companies. They really bring forward innovation. And once they are successful,
they take over then.
The large one take them over, buy them, they have money enough, and they have the innovation. Look at Pfizer, Moderna, Pfizer, BioNTech. That's a typical example, and there are numerous examples of that. And if the small company fails, bad luck for the small company, not for the big one. So they have just transferred the R&D risks to the small companies. But the nice thing, on the other hand, is when you're working on innovation, you do it in a nice, small environment, not a huge pressure from larger companies with all their timelines, etc. You can do it in your way. And then if you're successful, well, you can sell your invention for a lot of money, or the patent on your invention for a lot of money, or a good royalty deal to the big companies. It's like we did with the inhalers and some of our sugar glass technology. We sold that, or we licensed it out to companies. Or you can start up your own spin-out company, like we did with the Twinster and the Cyclops, where we have Pure IMS, which is a small startup company that we started for those inhalers. These ones were brought on the market by companies like Almirall, Forest, Mylan, AstraZeneca had a major impact. They sold probably most of the product so far. So, and that was through a license deal. So there are a lot of options to valorize your inventions, and that's great, of course.
Talking about Pure IMS, I was going to ask you about that. How I was interested. How were you able to manage your time? as an entrepreneur and as a scientist? They are both very demanding.
Yeah, certainly. And I must be honest, pure IMS could only be set up and be started because there were other people doing that. It was not me who started the company. I was involved, of course, and our knowledge went into it. And of course we pushed and we helped, etc., etc. But there were other people who were really starting a company. As a professor in a university, you should perform research. And of course you can help the people that are starting up the company using your knowledge. But it's not something, the business development, setting up production lines, market research, it's not something you should do as a professor. It's not what I'm good at.
Yeah,
I'm good in
Yeah.
performing innovative research. So this is really teamwork. Again, like we do all the research together with the people in my department. Here, you do the actions together with the people in the startup company.
Yeah.
And that may be a small number. With pure RMS, it still is a small number. Something like 7 or 8 people work there. So it's small, but still, they have to do the job. And I will help them. We will always help them. But we want to do their job. That's it. That's impossible. I have a 40-hour appointment at the University of Groningen, which in generally turns out to be a 60 to 70-hour appointment.
Wow.
But that's because I enjoy the work so much, so that's no problem at all. But then you are not able to do the company next to that too. It would even be highly unfair, because I'm paid by the university and I'm not paid by the company.
Yeah.
So what I could do is to quit at the university for 2 or 3 days, and then I could give more attention to the company.
Yeah.
But as long as I'm fully employed by the university, I should not do work, at least not a huge amount of work for the company.
Yeah.
And so you have to split that up. I'm a scientist. I'm not an entrepreneur. Probably I'm entrepreneurial.
Yeah.
I know how to develop patents. I know what you have to do to license them out. Also with help of others. It's not what you're doing yourself. You have to find help of others when you're outside your own strong points.
Exactly. So
your own
basically,
fields of expertise.
yeah, you should first innovate, have an idea, and then you could talk to others how you could maybe translate this into a marketable idea.
Yeah. So only through the help of other people we are able to do this.
Exactly. And maybe now there are students, medicine students, pharmacy students watching us. How do you see, do you see the chance of pharmacists, medical doctors, and scientists entering the entrepreneurial world, or is that still difficult for the next generation?
No, I think they should. More and more we see that the financing from government is reducing. We get less money. So if, as an academic, you want to have a good financial situation and be able to fund your own research, then this is one of the great ways to do it. You need to get money, to make money out of your own knowledge. And you can do that through having a fee-for-service company. You can do that through probably licensing out patents that you have founded, starting up companies. So those are the ways that you can make money to fund your own research. And it takes time, it's difficult, it's tough sometimes even. Also, we had failures before becoming successful. The chance that you are successful is very small. Never forget that.
Exactly. Yeah.
The number of patents that reach the break-even point is about 1 in 300. The number of patents that really make money is about 1 in 3,000. So we were extremely lucky. Next to the fact that we had to work quite hard for it, we also had some luck.
Yeah.
Be sure about that. But it's one of the ways that works. And there are always chances because we're in exciting times. We see fast developments. In my time, in the, let's say, the almost 40 years that I work now in pharmacy, starting as a student, ending up as a professor, and probably as a too old professor, I have seen pharmacy changing from something that was in many aspects considered to be an art into something that's 100% scientific. It's probably one of the most exciting scientific fields that we have today. As I said before, the interaction of a product, a chemical product, which is formulated into a dosage form with a complex biological system like the human being, it's extremely challenging, and we are starting to understand more and more. We have new technologies that can improve therapies, that enable us to administer drug a lot better, that even enable us to generate therapeutic effects which are even impossible without the advanced dosage form. So this really is exciting, and there are new methods nowadays. After the genomics and the proteomics, we are now entering entire mapping. We are doing gene mapping as a standard procedure. We have artificial intelligence helping us a little bit better to predict what's going to happen to drugs. So it's still extremely exciting what we're doing and challenging. So what else can you wish when you're a scientist? Not much more, to be honest.
Yeah. Out of curiosity, do you see the field, or do you see AI coming also to your field for biopharmaceutical technology at this point?
Oh yeah, for sure. Yeah, because it can help us to set up studies, developments, and it's not that big at this moment.
Yeah.
But in the future it will be. If you have the options here.
Somebody has to make it big.
Yeah. Well, they are making it big. It's becoming big almost by itself. 1 year ago, probably, I never used ChatGPT.
Yeah.
Now, whenever I have to write something, I start with
ChatGPT advice.
Yeah.
Why not?
Yeah.
It's there.
Exactly.
And it's pretty good. And, well, if I now have to design a new inhalation formulation for a specific drug or antibiotic or vaccine, I start from my head.
Yeah.
Probably in 5 years I will start
From the
from
computer.
ChatGPT.
Yeah.
Of course, it can only look back. So it cannot design innovations. That's what you still have to do yourself. But as a starting point, as a means that can help you to predict outcomes, it's very valuable. Still, we need a human creative mind.
Of course.
Exactly.
It's inevitable because there's no creativity in ChatGPT.
Yeah.
Not at all. But as a starting point, preventing that you forget certain aspects, it's very helpful. Artificial intelligence.
I guess a final question that we
Yeah,
also
we
ask
have that
most of our
tradition.
professors.
We have that tradition here in this podcast to ask always this question. So, what is, in your opinion, is the next $1 million question within your field?
Or in this case, multi-billion.
Multi-billion,
sometimes.
Multi-billion
I would
in
say so.
general,
Good
yeah.
point, Oscar. Yeah.
Yeah, in pharmacy, if you look at the amount of money that is made in drug, it's ridiculous. Keytruda was developed actually in the Netherlands originally, and now it's sold by an American company, unfortunately. 20 billion a year.
Wow.
So it's huge. But
It's insane.
well, what's the next $1 billion question? I think it's not just one. It's a multitude. We have
Interesting.
all kinds of new therapeutic options. The mRNA that came up, but also other gene therapies, ATMPs, advanced therapeutic medicinal products, as they are called. So cell therapy, extracellular vesicles. So there's a lot of new therapeutic options coming up. Well, what can the real added value in therapy be? What are the challenges for administering those drugs? Because administering a extracellular vesicle or even a cell, a human cell, requires quite different technology from administering aspirin or paracetamol.
Exactly.
Or diazepam. So we need really new technologies on that. So the question will be, what are the requirements of those new drugs? And how can we solve the problems related to that? And that's an ongoing question, because today we know pretty well how to administer the monoclonal antibodies, the therapeutic proteins that we have. 25 years ago, that was still a challenge. So there's an ongoing challenge, because we get new products, we get new challenges. And which one will be the biggest then? Maybe mRNA, maybe a cell or a cell fragment, or whatever it will be, or still one or the other very challenging protein, and whether it will be an endogenous protein, a protein that we are already carrying, or an exogenous protein that we have engineered. I cannot say that, but those are really the challenges that we are facing, which make also, in my opinion, our life really exciting.
Yeah, exactly.
Because solving such problems
is intriguing
can give
and
you
interesting.
and it gives you a lot of satisfaction if you. It's really rewarding to be successful with that.
I think so. Well, then there are a lot of possibilities, and that's very good news.
Yeah.
Well,
in pharmacy, there's always job security.
That's good. Well, Professor Frijlink, it was a pleasure. Thank you for the valuable knowledge, and thank you for being with us.
It was a pleasure for me too.
Yeah.
Thank you for having me here.
Thank you.